Well, thank you very much for being here. It's for me an honor to present Professor Andreas Nieder, the speaker of today. Andreas Nieder is a professor of animal physiology at the Department of Biology at the University of Tübingen in Germany, where he also serves as the director of the Institute of Neurobiology. So it has the same name that our institute. So he's an author of the book, A Brain for Numbers, The Biology of the Number Instinct. So this is a very top-of-the-notch neurophysiologist that started as a biologist. He was formed in the University of Munich, and then he did his PhD with Hermann Wegner in the University of Hagen. Then after that, he moved to MIT with Earl Miller, where he started his brilliant career on trying to understand how the brain represents numbers. Numbers in the parietal prefrontal circuit of monkeys, and then he jumped to humans, and he also jumped in a very creative fashion, as you will see, to crows. So he's a guy that likes comparative neurophysiology. He's a very quantitative, very creative, very thorough researcher, and as we will see, he's also a great speaker. So please welcome Andreas Nieder.
Yes, so can you hear me? I hope it's okay. No? But it should be on. Yes.
Maybe song, but very low.
It's better? Getting better? Okay. Let me know if I'm too low in my voice. I have a tendency to do this. So I'm very grateful to be here in Mexico, and I'm very grateful to Professor Hugo Merchant for inviting me to come here. It's very, very special, and I'm enjoying myself tremendously. I've seen beautiful work already in the morning, and I'm sure I will see much more. I would like to talk about the representation of numbers or numerosities, set size. So the question how many and how this is represented in the brain. And as you see, based on the title on these pictures here, I'd like to take a comparative approach. I'd like to talk about macaque monkeys. Last common ancestor between us humans and the macaque monkeys lived about 30 million years ago, which evolutionarily speaking is relatively recent. And the consequence is that we share a lot of anatomy and physiology with these monkeys. However, we are the only symbolic species. So only we can represent numbers in a symbolic way. Monkeys not yet. And this would be a human brain, and I will show you later some data from human single cells recordings in patients. And 15 years ago, we included another animal species, which is sort of the alien here, which is a bird, a crow, belongs to the family of the corvids. It's a songbird. Many people are surprised to hear that crows are songbirds, but they are. And the last common ancestor between these crows and us lived about 320 million years ago, which is about 5 times longer than the extinction of the dinosaurs. And from an evolutionary point of view, it's really an eternity. The consequence is that these birds, in their telencephalon, their end brain, have a very different design, a very different layout. And this is interesting for guys like me, who is trained as a biologist and a zoologist, because this allows us now to investigate what are the differences and commonalities between vertebrate brains that allow all of these smart animals to represent numbers or numerosities. This is the menu for today. I will start with monkeys, basically for historic reasons, because they've been the first species where we gain some insight. And I will talk about number selective neurons, neurons that are really specifically tuned to a specific number that the monkey has to deal with, and then switch over to the question, how is it that monkeys can represent quantitative rules, such as greater than, less than? And I will come to this in a second. Then the middle part is about humans, and I guess we all are very interested to learn something about our own brain. I guess at the end of the day, this is what is driving us to do neurobiology, even with animals. And a while ago, I was fortunate enough to team up with an epileptologist from the University Clinic in Bonn. This is Florian Moormann here, and I will talk about this in a second. But this allows us now for a few years to also gain insights into how our own brain is representing numerical quantity, and then not just non-symbolically, but even symbolically, real numbers. And then finally, I switch over to crows and tell you a little bit about how crows represent numbers, but then not only from a sensory point of view, which is the main part of the first two chapters, but also from the point of view how can they self-generate a specific number of events. You may ask, what's the purpose of investigating animals in terms of numbers and in the number theory? Because I just said they are not symbolic. They are not really representing abstract numbers, but they can represent the quantity of objects in a set, what we call numerosity or set size. And I argue that this is the precursor representation for everything that's coming later in us humans that allows us to represent real numbers in a symbolic way. And there are at least 3 lines of evidence why I think this way. One is coming from anthropology. There are courageous people that go out into the wild, into regions around the Amazon, the equator. This is a world map, and you see the red dots here. These are indigenous people that have been studied for their capability to represent numerical quantity. The fascinating thing about many of these hunter and gatherer societies actually is that they have not evolved a counting system. They cannot count like you and me, 1, 2, 3, 4, 5. It seems hard to believe because it seems so natural to us, but when you think back as children, this is something we had to learn painfully over years. So if you live in a culture where no one is teaching you to count, you are left with this ancient system to non-symbolically represent set size, the number of objects in a set. And this is what these people also can do, but they show some systematic errors as soon as the numerosities become higher. And I'll show you in a second how these errors look like. And they point to the fact that we have, all of us, when we are born, we have an analog number system ready in our brain that allows us in an approximate way to represent numerical quantity. A second line of evidence, very interesting one, is coming from us humans, from little infants. Developmental psychologists tell us for many decades now that even these little infants at the tiny age of a few months can discriminate between set sizes. These little infants cannot be taught to tell us anything, so the psychologists resort to spontaneous behaviors, habituation, dishabituation protocols, and the like. And the youngest subjects that have been tested so far that I'm aware of were infants that have been investigated 50 hours after birth in the hospital. So they can hardly look straight, if you know little infants, but even they can discriminate set size, and not only within the visual domain, but even across the visual and motor domain, the visual and the auditory domain. Which strongly emphasizes, again, that there's something residing in our brain that allows us from birth, basically, to discriminate set size in an approximate way. And very interesting, there are many data out there by now that tell us that the precision with which these infants can discriminate the number of objects in a set is predictive of their capability to perform true mathematics in school later. So the performance score for mathematics is higher if, as children, they have been able to discriminate these set sizes more precisely. And that tells us, I guess, very directly that there's a link between this non-symbolic and the symbolic representation of numbers. And the last line of evidence for us, of course, as biologists, very important. There are multiple studies out there that tell us that also animals, without a scientist like myself around training these animals, are using numerical information to arrive at informed decision and to have survival and reproduction advantage, which, based on biology, is all that matters. A while ago, I made a survey across the literature to find out where we find such examples. I'm not going through all of this, but I guess it's immediately clear that an animal has an advantage if it is able to discriminate more food items from fewer food items in order to pick more food items to survive. In cooperative hunting, for instance, it's known that wolves, for instance, team up in specific numbers in their pack to hunt prey. They team up to around 7 of 8 or 8 members in their pack when they hunt deer. But if they hunt the most formidable prey, such as bison, they team up in about 13 animals. So they have an optimal number of a hunting party. Fewer is bad, more is bad. So it tells that they have some idea of numerical quantity. And again, I'm not going through all of these examples, but there is ample evidence that it matters to animals spontaneously. It has an adaptive value, as we biologists say. So I start now with our monkey work, introducing the task that the monkeys are being trained on. Everything we do is based on behaviorally trained animals, very similar to the brilliant work of Dr. Merchant. And in our case, we are interested in numerical quantities, so not in a continuous quantity like time, as in Dr. Merchant's case, but in our case, the discrete numerical quantity. Our animals typically perform something that we call a delayed match to sample task that goes like this. So imagine a monkey sitting in a dark booth, is facing a computer monitor, is grabbing a manipulandum to indicate to the computer that now it wants to work. And in addition, it has to fixate on this little fixation dot in the center of the screen. If the monkey is doing this, a sample stimulus appears, cueing the animal for a certain number of objects. In this case, the number 4 would be the important number. Then a delay period appears without dots on the screen. This is a classical working memory period where the animal has to memorize the number of dots that it has just seen in order to solve the task, because later it has to match the number of dots with the number of dots in the test stimuli. And in 50% of the cases, after the delay, a match stimulus appears, showing the same number of dots, in this case 4, and the animal has then to respond by releasing the manipulandum to indicate to us, now I've seen the number 4 twice in a row. In the other 50% of the cases, we show, however, a non-match, showing fewer or higher numbers of dots. So 3 here would be fewer than 4. And in this case, the animal should maintain the lever and wait until the second test appears, which then is always a match, and then to release to get a reward. It is obvious when we have these kinds of stimulus layout that there are also parameters, visual parameters, changing inevitably when we change the number of dots. For instance, if you have 2 black dots and you double them to have 4 black dots, you're also increasing the area of blackness on the screen, which is a purely perceptual parameter that has nothing to do with number. And animals might be very sensitive to this parameter and then do the task according to the blackness on the screen, not the number of the dots. So we have to control for that. And I'm not going into the detail. I hope you believe me, we are very aware of these problems. We have a range of different control stimuli that we show together with some standard stimuli in the background, unbeknownst to the animals, where we control the area or the circumference, the density of the dots, and so on and so forth. We have shown when the monkeys are well trained, they do not care about these changing non-numerical parameters. All they care about is the number of dots on the screen. And again, I'm not showing you the data. I hope you can believe me. You can read it up, of course. And how do the animals respond to this task? Here I'd like to show you the discrimination performance of the monkeys, resulting in what I would call a representation, a behavioral representation of numerosity, of set size, for these animals. You see here the response frequency of the monkeys plotted against the number of items that we show. So we not only show the number 4, but we show a variety of different numerosities. From trial to trial, these numerosities may switch, and in every trial, the animal has to see and memorize a new numerosity. This is a data point when 4 is the match numerosity, similar to the display that you've seen before. And the test stimulus is also a 4. So these are the so-called match stimuli. And you see in about 90% of the cases, the animals release the lever and tell us 90% of the cases they detect 4 as being 4. More telling, however, are the non-matches. So when we have a deviant numerosity and the animals are responding, and this is depicted here. So if we show 3 as a non-match, the animals tell us in about 60% of the cases that they would perceive 3 as being equal to 4. And if we show 5, then the animals tell us in about 70% of the cases that they would perceive 5 as being 4 dots. So here they make a lot of errors. And this is why it's called an approximate system. It's not very precise, it's approximate. But you see when we move the deviant further away from the sample stimulus, 2 and 1 and 6 and 7, then the errors are decreasing. The animals are becoming better until they are almost perfect. And this results in what I would call this behavioral representation of the monkeys for numerosity, in this case numerosity 4. And you see, because this is a bell-shaped function indicating this imprecision, it gives rise to one of the very well-known phenomena when dealing with numerosity, the so-called numerical distance effect. Relative to a given target numerosity, when we increase the distance of a non-match, performance gets better. And this is a very classical phenomenon that can even be seen when we count symbolically. We are not only showing numerosity 4, but target numerosities in this experiment from 2 to 6 here. And you see for all of these target numerosities, we get these bell-shaped performance functions. When you look closely, you see that the performance functions are becoming broader the higher the numerosities are becoming. And this is not an accident. This is something we see for many animals across time. And this is giving rise to the so-called numerical size effect, meaning that at a given numerical distance, between two numbers, it's becoming progressively more difficult to discriminate them if the absolute values of the numbers are increasing. And this is giving rise to this broadening of these performance functions. If you look very closely, you may even see a third effect, and this is based on the phenomenon that these functions are not entirely symmetric on a linear number scale. So you may see that the flanks to the lower numbers are more steep than the flanks to the higher numbers, more shallow. And this is again a systematic finding we make, and this is giving rise to so-called Weber's law, meaning that the representation of numerosity follows a logarithmic number line. If we take exactly the same data and plot them on a logarithmically scaled or logarithmically compressed number line, then the performance data become very symmetric. Again, indicating that the original number line that animals, and I would argue also we carry in our brain, is a logarithmic number line. It's not yet a linear number line. This is something that comes later when we are trained in school to deal with numbers. Yes? Yes?
Number 0.
Yes, number 0. Very, very fascinating. Unfortunately, I have not included it in my talk because it's an entirely separate other talk. The animals can represent 0 when we train them, so they understand the empty set. And we find also many neurons, I'll come to this, the neurons in a second, that are representing the empty set. I would be happy to say more about this in the discussion. Unfortunately. Yes, please.
I have another question about distribution of the pattern of the dots. The pattern that in which you present the
Yes.
dots, it's important that
The pattern,
for
so
identification if they are like in a cluster or distributed around.
yes, okay, that's a good question. So all of these stimuli are generated by MATLAB programs. So in order for us to control the parameters that we have in them, the patterning should not matter to them. You may have seen that the sample stimulus and the test stimuli, they are always different. So the animals cannot perform the task based on the pattern matching of the visual pattern. Now, we train the animals in order to then learn something about the brain, The two areas we classically record from in macaque monkeys are the prefrontal cortex and the fundus of the intraparietal sulcus, an area called VIP, ventral intraparietal area. Both of these areas get input from the visual modality based on the dorsal visual stream, as you can see here. And the reason why we are focusing on this area is that this seems to be the core area of the number network, not only in monkeys, but also in our human brain. I'll come to this in a second. And we typically record from these two areas simultaneously, as I said, single unit recordings with multiple electrodes in these areas. And what we find, fortunately, made us very happy, are indeed neurons that are specifically tuned to one or the other numerosity that the animal has to discriminate. And this is one such a neuron. I briefly guide you through what you can see here. The top panel shows a dot raster histogram. Every line is a trial. Every dot represents a trial, and the x-axis shows the time, and every dot represents an action potential. The time here is the time throughout one single trial. 0 would be the start of the fixation. Interval 500 would be the start of the onset of the sample numerosity, and after 1,000 milliseconds, the delay period, the working memory period, without dots on the screen would appear. And here we have presented numerosities from 1 to 30. We didn't get any higher because it's getting very nasty then to control for non-numerical parameters. But the idea is that the monkeys and the neurons could represent even higher. And I hope you can appreciate here for numerosity 2, we have this blackening of the individual dots, indicating that the neuron is heavily firing whenever the monkey sees 2 dots, and less so when the dots are increasing or when it only shows 1 dot. The same kind of data are integrated and displayed in an averaged way in the spike density histogram. Here the firing rate of the neuron is plotted against the time, and the different colors indicate here some of the numerosities that we've shown to the animal. And I hope you can see that this neuron really has the highest firing rate whenever we show 2 items. That's the blue curve. The second highest would be for 4. I hope you can still see this, the cyan curve here. And then we have 6, 8, and so on. So then gradually the discharge rate is decreasing. And 1 would be the black. This is also a very high firing rate. So the neurons are showing something like the numerical distance effect, but now on the neuronal level. And we plot these data then in terms of tuning function, where we have the firing rate plotted against the number of items. And you see here the peak here for numerosity 2. This indicates that this neuron is most interested in numerosity 2, and then progressively less for more distant numerosities. So we have a real tuning to these numerical quantities in the monkey brain. And for this neuron, it's also highly interested in the numerosity 2 during the delay period, when the animal has to memorize what it just has seen. So this is a neuronal correlate for working memory for numerical quantity, if you like. One other example of a neuron that is beautifully tuned to numerosity 6 in this case, and then progressively drops off for the other non-preferred numerosities. And for any numerosity we show the animals and they have to work with, we find certain neurons that are specifically tuned to these numerosities. These neurons have, a journalist once called it the beloved number. I like this expression, so that's why I'm telling you. Now, these data that I showed you so far were all for the simultaneously displayed dot patterns, which in a way is maybe not the typical stimulus or something we typically understand as counting. When we talk about counting, we usually mean a sequential process because we count 1, 2, 3, 4, 5. So it's something that happens over time. And at some point we were wondering, could we also find such neurons when we teach the animals to enumerate dots and sounds over time? And this is the protocol that we used here. So you see, instead of multiple dots displayed at once in the sample period, we have 1 dot in a row, segregated by no dots in between, a delay period, and then matches and non-matches as before, and the monkey has to tell us whether they have seen 1, 2, 3, or 4 dots in a row and then match them to these simultaneously displayed items. I should mention here that in contrast to Dr. Merchant, we are not interested in representations of rhythm or timing or anything. In fact, we destroy these parameters by engaging different protocols that I'm not showing you, but we have different protocols that are so arbitrary in their timing that the monkeys cannot use time as an information to solve the task, because we are interested in the number representations. In addition, we managed to train 2 monkeys not only to represent the number of dots in a row, but also the number of sound pulses in a row. And people working with monkeys know exactly that this is something that comes very hard to the animals. Fortunately, they did it. So here, for instance, if they hear beep, 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 beep in a row, then they know they have to match it to 4 dots afterwards, after the delay. And this is something the animals learned, fortunately, and provided us with the opportunity to record. And we were then interested whether they would show activity for the first, second, third, and fourth dot that we represent. And what we found in the prefrontal cortex specifically are neurons that are still nicely tuned to 1, 2, 3, or 4 dots in a row in the sample period, and you see some here. And they were however, not interested in the sensory modality. So whether we presented visual dots or acoustic sounds didn't matter to these neurons. All they cared about was the number of events that was presented. And here you see the normalized activity plotted against the number of items. We only presented 1 to 4 items. And you see, based on the color, 4 different neurons. The solid lines indicate the sound presentations. The dotted lines indicate the firing rate for the dots, the visual presentation. And based on the statistics, we tested all of these neurons with multifactor ANOVAs. They did not discriminate between the visual modalities. All they cared about was the number of items on the screen. This is something we found in about 20% of the neurons in the prefrontal cortex, the highest integration center in a primate brain, I would say. We did not find it in the intraparietal sulcus, which is a little puzzling, but I'll come to this in a second. So number selective neurons for sequential and multimodal representations are present in the monkey brain, and these are very, very abstract encoders of numerical quantity because they do not care about time or space or the sensory modality in which these items are represented. We recorded from different areas over the different years. This is a depiction of a lateral view of a monkey brain. Left would be anterior, and you see these colored areas here. These are some of the areas from which we recorded from over the years. The redness indicates the proportion of numerosity selective neurons in an arbitrarily selected set of neurons. So we take all the neurons that have a good signal-to-noise ratio and analyze them, and post hoc determine whether they are selective for numerosity or not. And you see in the fundus of the intraparietal sulcus, this is the intraparietal sulcus unfolded so that we can look into the sulcus. We have a relatively high proportion of about 20% of the neurons that is interested in numerical quantity. This is the area VIP, ventral intraparietal area. And in addition, we find usually even more neurons representing numerosity in the prefrontal cortex. When we have these 2 hotspots in the brain, and you see the other areas in the posterior parietal cortex or areas in the inferior temporal cortex are not interested in numerical quantity. If we have these 2 hotspots, then an obvious question is, what is their relative contribution to extracting numerosity? And it turns out for several monkeys that we've investigated so far is that the neurons in the intraparietal sulcus are representing numerical quantity earlier compared to those in the prefrontal cortex. So our current working hypothesis is that this is really the first stage in a primate brain where numerosity is extracted, and then it sits there and waits whether this information is needed for certain types of goal-directed behaviors, as in this delayed match to sample task that I showed you. And if it is needed, it is probably routed forward to the prefrontal cortex, and usually also a little emphasized. We find more neurons, and the neurons there are also a little more selective. The question is, of course, now, does this have anything to do with what's happening in our brain? After all, we are recording in a non-human primate, so hopefully this is somehow related to what's happening in our brain. And with this slide, I would argue that indeed it has a lot to do with what's happening in our brain. This is here functional imaging data, so not single cell data, but blood volume changes usually, MRI data, graphically inflated human brain, left hemisphere, again left would be anterior, the eyes would be around here, and the yellow patches here are the areas where whenever we humans represent numerosity or numbers or count or do mathematics or mental equation solving, in all of these cases, based on these meta-analysis of about 30 studies, parts of the posterior parietal cortex and parts of the prefrontal cortex are becoming activated. And if you compare this to a non-human primate brain, these are, I'm pretty sure, homologous areas. These are exactly the areas that we find in a non-human primate. A more direct evidence comes from lesion studies. Neuropsychologists are investigating performance in people that, based on some insults in the brain, show behavioral deficits. And here you see an insult, this gray shaded area, around the intraparietal sulcus of a patient that has been studied. And such patients typically develop very specific deficits in dealing with numbers and numerical quantities, even though the language system is entirely spared. It's very important for me to mention this because many people think that counting is a variant of language, but it is not. It is really a separate system in our brain, helping us to represent number and to do mathematics. Yes?
I think neural tuning to numbers is in fact the result of a very complex processing by the neural. So I imagine that previous to this tuning in some cortical area, the stimuli, for example, have to be first. The subjects have to identify each different item, each different dot as an individual element. Then to compute and approximate in order to get that tuning to the different numbers. And regarding the simultaneously presented number of dots, the other case where the dots appear in sequence, then probably for each dot it has to be done accumulation or something like that. Do you have some data or do you have some idea of how the process previous to the neural tuning occurs in the cortex or in which cortical areas is during this process?
Yes. The very short answer is no, we don't. You're absolutely right. There has a lot to be happening before we find such neurons. So our approach is, if you like, a top-down approach. We start at the very apex of the processing hierarchy in the brain, prefrontal cortex, posterior parietal cortex. And now there is a need to work down our way to the more visual or more sensory areas to see exactly right how these items are individualized based on the receptive fields and so on and so forth. But I cannot present any data. I don't know exactly how this is done. All I can say that these are veridical representations of numerosity, these tuned neurons. We've seen them over and over again in different areas of the brain. And I think there's something like the output stage of the system. It may be that at a precursor system, pre-stage level, there are some monotonically tuned neurons, but we do not find them with our tasks. I know there are networks that assume such summation units, but we usually do not find them. Yeah. Please.
I imagine that there can be tuned curves for every possible number. At some point, tuned curves must stop being the way to represent this. Do you know, is there a particular limit at which this starts becoming?
Yes. So, okay. Also, very valid question. If we go to higher numerosities, are there still neurons that are representing them? I would argue, based on what we've seen for numerosities up to 30, that there would still be one or the other neuron that has something like a preference for, I don't know, maybe around 100 dots. But I'm clearly saying around 100 dots. The functions are, like the behavior, the functions are becoming increasingly broader, less selective. And then at a certain point, it's a matter of taste, basically, where you say is the preferred numerosity. All of this follows very nicely Weber's law, but it is the same in all sensory modalities with intensities. And I guess there is no theoretic reason why this should not be possible up to a certain limit. Of course, at some point, everything becomes really a lot, and a monkey could not be able to represent 255. That's, of course, clear. But until a certain level, I would think there are indeed such neurons present. I should also mention that not all neurons in these areas are representing numerosity, and I'm also not claiming that this is exclusively what these neurons are representing. And in fact, we've seen this if we train the monkeys on different tasks in the same session. We see that some of these neurons also take part in other tasks. So these neurons, of course, are working in ensembles, in groups of neurons, and the population matters, of course, a lot, not just a single neuron. Now, I'd like to take you one step further. So far, I've shown some representation of numerical quantity, but this in itself is not very helpful because the animal needs to work with this representation. For instance, in arriving at decisions, fewer opponents or less opponents, and then either attacking or retreating. These would be some natural decisions an animal in the wild would make. So we thought, is it possible to find neurons that would represent rules, even switching rules between such quantitative or mathematical operations? And one of the first mathematical operations we learn in school is a greater than, less than operation. Maybe you remember this in the first grade. And this is something a monkey can also still manage in an approximate way, and we managed to train animals on such a task. So here you see an elaborated version of the delayed match to sample task. Monkey is fixating, sees a certain sample, numerosity 5, is remembering it, then a rule queue appears, which is the red circle, and the red circle indicates now pick a numerosity larger than the one you have seen in the sample. So now pick a numerosity that is greater than 5 in this example. Then after a second delay period, and this second delay period is very dear to us because we call this a rule delay period, because the animal now has the rule available, what to follow, but it cannot prepare yet a motor response because it's lacking the information. And then comes either a match, which would be 8, I think, dots. The animal has to respond because 8 is larger than 5. If we show 3, the animal is not allowed to respond and has to wait for a while because 3 is smaller than 5. So this is what I would call the greater than task. And of course, we also have a less than task. Pick the numerosity that is smaller than the sample, indicated here by a blue circle. And again, for us, this period, the second delay period, is the one that's most interesting. And we are not only showing numerosity 5 here, but different numerosities. And according to these different numerosities, the animals have to very flexibly make a greater than or less than rule switching task. For a monkey, this is pretty challenging. From a behavioral point of view, we would be ready to go. But from a neurophysiological point of view, not yet. Because I just said many times we are interested in what's happening in this delay 2 period. And you see we have a confound here, because the rule is confounded by the color of the rule queue. If a neuron would respond in this period, we would not be able to say this is because the animal is remembering red, or because the animal is really remembering the abstract rule, follow the greater than rule. And it's the same here. So the poor monkeys had to learn a second set of rule cues, a white circle with some drops of water, a white circle without drops of water, indicating also the greater than or the less than rule. And now we have the opportunity to take these 2 rule cues as factors into our ANOVA and make sure that whenever a neuron is responding, it is responding only because of the rule and not because of the appearance of the rule cue. And as I just said, we are not only presenting numerosity 5, but 2, 3, 5, 8, and 13, and relative to all of these numerosities, the animal has to make a greater than, less than decision. The animals can do this. This is behavioral performance correct. Across the sample numerosities, you see on average they perform at about 80% correct, relative to a chance level of 50% because we have two choices, so this is highly significant. And most important, this is one of our beautiful neurons that we call rule selective. You see again a dot raster histogram and a spike density histogram. The important period, as I just pointed out, is this delay 2 period that you see here. And this neuron increased its activity, shows this ramping activity, whenever the animal is preparing for or following a smaller than rule, not when it is following the greater than rule. And you see this increase in activity for the two cool colors. So blue and green indicates both the less than rule, while the warm colors, orange and red, indicate both the greater than rule. And you see, and we did the statistics, obviously, the neuron does not care about the rule cues. All it cares about is the rule, whether it's a greater than or a less than rule. And this is one of the reasons I am personally in electrophysiology. I think these are extremely abstract signatures of neurons for an abstract rule that they are following before they can prepare any motor act. So an abstract principle, if you like, that they are following. And we are not only finding one of these neurons, but about 20% of the neurons in prefrontal cortex show this rule selectivity, and it's about half and half. Half of these neurons are representing the greater than rule, and the other half are representing the smaller than rule. Now I switch over to my second chapter, which is dealing with human data. And I just mentioned the heroes of this work is my colleague Florian Moormann, epileptologist at the University Clinic in Bonn, and our PhD student is the Kutter. And the reason why we can do these recordings is that Florian Moormann is an epileptologist, and he's treating patients and he's trying to help them. And some of these patients take medication and are becoming seizure-free. But for other patients, the medications do not work, so they may decide that in a surgical intervention, the focus of the epileptic seizure is removed from the brain in order to help them to get rid of these seizures. Now, interestingly, even with the great non-invasive methodologies we have available, it's not possible to tell where exactly or in which hemisphere of the brain the seizure emerges or the seizure has its origin, and that's something the neurosurgeons need to know. So in a first intervention, there are electrodes, several electrodes implanted into the brains of these patients. Then they wait for a few days in the patient bed until the diagnostic measurements are completed. And then in the second intervention, the focus of these seizures is removed together with the electrodes. While they're sitting in the patient bed, waiting for the diagnostics to be completed, if they are ready and if they give us their written consensus, we can test them with different protocols. And in our hands, of course, numbers and mathematics is very important. And you see that they can work with laptops where they do certain tasks, similar to what the animals can do, but of course more because they can really count and understand true numbers. These are the types of electrodes we are using, the typical Fried types of electrodes. These are the diagnostic contacts that you see, and these will be the electrodes that allow us to record single-cell activity. Many of them are being implanted into the brain. This would be an MRI section here, where you see a horizontal MRI section where you see the electrodes implanted. Almost always it's the medial temporal lobe that is causing the problems, which is why almost always these electrodes are implanted into the medial temporal lobe. These are areas like the hippocampus, parahippocampal areas, and so on and so forth. And then, as I said, we can start to record if we have the written consensus of the patients. That's a brief layout, functional neuroanatomy. The hippocampus is the highest level in this medial temporal lobe, but we are also interested in the entorhinal cortex, parahippocampal cortex, and the amygdala, which is not part here. And yeah, that's the medial temporal lobe in the human brain. Before I was telling you that the posterior parietal cortex and the prefrontal cortex are the core number areas in a primate brain. Now I'm switching over to the medial temporal lobe, areas such as the hippocampus. Why is this? Is this justified? On the one hand, we have to do it because we are working with patients, and as I just told you, usually the problems are emerging in the hippocampus in these areas. That's why we have the electrodes there. But there's also more and more evidence that the hippocampus or the medial temporal lobe areas are in fact involved in doing mathematics. In children, for instance, it's more and more known that when they learn, for instance, multiplication tables, that there's huge activity in the hippocampus, indicating there's number representations also in the hippocampus. So it's not sort of an irrational idea to assume that we would find neurons in these areas. One of the tasks we had the patients perform is a very simple task. It's a simple parity judgment task, in this case with dots again. So here you, for instance, you see 5 dots, and is 5 dots odd or even? What would be your idea? So that's what parity means. Is it an odd or an even number? 5? It's an odd number, yes, exactly. And we represented numerosity from 0 to 9, and all they had to do whether is this an odd or an even number. And again, we made sure that the patients could not rely on non-numerical factors, but were really assessing the number of dots. And in fact, and that much to my delight, we found many neurons, 60% on average in these areas, that were significantly tuned to numerical information, very similar to the ones I showed you in the monkey brain. Here is a neuron that is tuned to numerosity. Let me see, yellow is 3. So here you see the highest discharge for 3. You have to be aware that the neurons in the medial temporal lobe are discharging very faintly only, so the firing rate is always very low. But we have the same, exactly the same type of code, these numerosity tuning that we have seen before in the monkey brain. And for me, this was extremely rewarding because this tells me that what we see in the monkey is really also relevant for our own brain. Here is another example of a neuron in the hippocampus, beautifully tuned to numerosity 7, as you see, and then showing this drop-off in this bell-shaped tuning function. We were working with numerosities from 1 to 9, and maybe some of you know that there's the idea out there that we are representing small numerosities differently compared to larger numerosities. And by small numerosities, I mean numerosities or numbers from 1 to about 4. And people call this a so-called subitizing range. Subitizing comes from a Latin word meaning that we very rapidly arrive at this decision, and it's very a lot without errors. But when we are dealing with higher numerosities, 5, 6, 7, and 8, then we start to estimate. We are using this approximate number system that I was alluding to before, and then become very error prone. And the first study that I'm aware of that showed this is from Jevons 150 years back. It's a funny Nature paper from the time because it consists of only his data, Jevons himself, and he was performing the experiment himself and then took the data from himself and was publishing a Nature paper. That's no longer possible these days, but in the good old days that was. So he took some beans he's describing very beautifully, and he threw them casually, as he says, into a paper box, and then instantly, without counting, assessed the number of beans in this box. However, he found something very interesting and something that's still valid today. So this is the actual number of beans represented in the box, and this is the number of beans he estimated to be in the box. And what you see, he always got it right for 3 and 4 beans. He never made an error. But for numerosities 5, 6, and higher, he started to make errors. And since then, there is this idea around that there might be a coding boundary, some representational boundary between the smaller and the larger numerosities. I never believed in this idea because I just never saw it in monkeys, but I have been convinced that there is probably something to it, because when we now look at our human data, we see a clear difference between the representation of the small numerosities and the large numerosities. For instance, when you look at, again, these data for numerosity that was again 2, and here it is 3. And now in addition, what I'm plotting here is the spontaneous firing rate, the baseline activity of the neuron. You see that these neurons go beyond the baseline firing rate before they are actually actively encoding their preferred numerosity, but they're not doing this for higher numerosities, such as 6 in this case, or 7 in this case, where they stay at around the same baseline firing rate. And we saw this very representative in all of the neurons, and for an electrophysiologist like myself, this is something to work with. This was very obvious. And when we plot these tuning functions of the neurons, we average them, normalize them based on a set score, and the set score simply means that 0 is always the baseline firing rate of the neurons. We see that for the tuning functions 1, 2, 3, they are then going below baseline activity for the non-preferred numerosities, but the tuning functions for higher numerosities, higher preferred numerosities, don't do this. So there's an obvious difference between these two. Then we characterize the tuning functions by fitting a Gauss function to the actual data and derived the amplitude of the Gauss function and the sigma as a measure of the width of these functions. And what we saw is that for the small numerosities, the amplitude hardly changes, but then there's a progressive increase in the amplitude for numerosities 4, 5, 6, and higher. And we saw something very similar for the sigma, for the width of the tuning functions. The width stays about the same for the small numerosities, and only from 5 and 6 on, it increasingly is becoming broadening. So that smells like a coding boundary. We investigated this in more detail by using the entire population of numerosity selective neurons in this case, and we performed something that is called a state space analysis. In short terms, the activity of the neuronal population is conveyed or translated into a spatial representation, and then based on the spatial layout, one can see coding differences. In other words, at any time in the trial, the activity of n neurons, 120, is represented by an n-dimensional vector in an n-dimensional space. And because we then have 120 vectors and spatial dimensions, this is something we cannot visualize anymore. So what people are doing is they reduce this amount of data to the 3 most informative dimensions, and this can then be plotted in a 3-dimensional space, and this is what you see here. We do this for every single numerosity that the neurons are tuned to, and we do this across a single trial. So this ends up in a trajectory that is meandering in the 3-dimensional space from the fixation to the sample representation to the delay and to the people's motor response. All of this is, again, very abstract, but the important information is when you look at the distances between these trajectories. And what I hope you can very clearly see is after the sample stimulus and the delay happens, we have a split up of these trajectories into 2 different groups. So the cool colors here, the bluish colors, are meandering here on top of this 3-dimensional display, and the small numbers, 1, 2, 3, 4, are meandering here on the bottom of the 3-dimensional space. So obviously there's a coding difference between these small and large numerosities. And we quantified this, of course, not just by looking with our eyes at it, but doing statistics. We took some sections in 2 dimensions between these trajectories, and then this is something what you can see here, dimension 1 and dimension 2, and we were asking, is it possible to find different clusters in these displays? What is the optimal cluster size in this display? It could be 1, 2, 3, or 5, could be any. So we were resorting to some statistical measures. I'm not going into the detail. 2 different ways of determining the optimal amount of clusters in these displays, and both methods told us the optimal numbers of clusters is 2. So there are 2 clusters that are really categorically different in these displays. And then the question was, where is the boundary of these 2 clusters? And based on an unsupervised k-means clustering approach, we found that the boundary is between 4 and 5. So there's something very different happening between 4 and 5, and this is really nicely relating to this idea that there is subitizing and approximate number representation in the human brain, and that's something we want to explore in more detail in animals in the future. And probably it's not an accident that in different human cultures around the world that independently involved counting notations, the first 3 or the first 4 notations are represented by the actual number of a mark. As you can see here, for instance, 1, 2, 3, 4. So you simply can assess the number of the marks, and only later for higher numerosities, actually I should resort to this here because this is the Maya notation, very highly developed. You see that we have dots up to 4, and beyond 4, for 5 and higher, we start with some more abstract symbols as you have. And this is probably indicating that we have an advantage to assess very rapidly the small numbers, and we don't need really symbols, and the symbols kick in only later in all of these counting systems. Yes?
I remember in the first slides you showed that for some cultures there is no symbolic representation number.
Yes.
In that cases, in those cases, these cultures lack also a symbolic language.
No, they have language. They have symbolic language. They just don't have a notation system.
Notation.
Yeah.
Okay. Yeah, because I think in this case, except from the first numbers, I think for the other symbols, I think in an arbitrary sensory symbol mapping.
Yes, of course, this is something we have to learn. Yes, obviously. But what I'm saying is we don't need to learn a lot for the small numbers. We can assess them very intuitively and very precisely. And this is obviously reflected in these notation systems. Thank you. So, but the advantage, obviously, working with human patients is that we can also not only investigate dot numerosities, but also real number symbols, Arabic numerals. So we had another protocol where we represented dot numerosities together with Arabic numerals. And here the task is more complex. This is a real addition or subtraction task. So here you see the plus sign, and the patients have to add 2 and 4 in this case and indicate to us that the result is 6. And in other trials in the same session, we present the same addition or subtraction problem with Arabic numerals. And that allowed us now to compare whether we may find neurons that would encode numerical quantity irrespective of the notation. We are again changing the layout of these stimuli in order to make sure that we are really measuring numerical quantity, and we do the same also for our rule, for our mathematical rule, the addition and subtraction. And I'm now focusing on this part here where we show the first operand and the delay period, whereas nothing else is happening in the minds of these patients. They do not yet know the calculation rule, so they are not yet calculating. All they do here is to represent the numerical value. And what we find is, again, neurons that are beautifully tuned to these numerosities on the left-hand side. And then the question is, would they also be tuned to Arabic numerals, and particularly to the same numerical value? Here is a neuron that's nicely tuned to the dot layout, non-symbolically, but it's not statistically significantly tuned to numerosity 3 or any other, sorry, not numerosity number. This is a neuron that is tuned to numerosity 3, but it is again not tuned to any Arabic number. This is one of the very few neurons that is tuned to 5 dots and is also tuned to the Arabic number 5. This indicates already to you that even though we find many neurons that are tuned to specific numerosities in these areas, and also significantly more neurons than expected based on chance that are tuned to symbolic number, we hardly find neurons that are tuned to both notation representations, which for me was very disappointing because I'm always looking for these abstract encoders of number. but we do not find them in the medial temporal lobe. So it could be that this is just my illusion that numbers and numerosities are differently represented in the brain, or it could be that in other areas of the brain, maybe in the posterior parietal cortex or the prefrontal cortex, this is happening, but not yet in the medial temporal lobe. That's something I would like to find out, but as I said, it's really not easy to record from these areas because these are human patients, and of course, we need to pay attention to their needs. Now, we have a calculation rule, so I go a step further. What is happening when we humans do real addition or subtraction in a symbolic way? And could this be represented by these neurons? And the answer is yes, very beautifully so. Here is a neuron, and this is, the timeline is again the task. Here is the calculation rule displayed, and this is then the delay after the calculation rule. So from this point on, one could expect a representation of an addition or a subtraction rule. And here you see a neuron that is nicely increasing its firing rate whenever the patients are adding for two different rule cues, again, as I showed you before. So the neurons are not discriminating between the sensory features of the rule cues. They're really discriminating selectively between the calculation rules. And here is an example neuron that is discriminating or shows a high activity for subtraction as opposed to addition. And we find about half and half. We were wondering whether the entire population of neurons now would be able to tell us or to predict an ideal observer, just listening to the firing of the neurons, whether the patients would do addition and subtraction. And here is from the parahippocampal cortex and the hippocampus, this were the areas that were mainly involved in the task, the accuracy of a support vector machine classifier, a statistical classifier that was fed with 50% of the data in order to learn whether at the moment when they have been shown the data, the patients were doing an addition or a subtraction, and then tested, and these are the data you see here, with the remaining 50% of the recordings that the classifier had not yet seen in order to predict what the humans would do based on the firing. And you see very nicely that the accuracy is way and significantly above chance level of 50% in the parahippocampal cortex and in the hippocampus. So these neurons do contain valid information about the calculation rule in the human brain, helping a classifier to predict this. And this is very briefly, also the classifier based on these data does not care about how the calculation rules are represented. All it cares about is what calculation rule is displayed. Yes?
So these are patients where maybe some of these areas that you recorded are already, reset. They have a bigger focus. So I'm wondering if the subjects then can add or subtract once the neurosurgeon illuminated the areas that you recorded.
Yes. That's actually a great question, and I do not know any paper that has investigated this. I'm not aware of a paper that has investigated bilateral hippocampus patients for mathematical capabilities. That's something we are lacking. That's one of the questions the reviewers always ask when we submit our publications. So I don't have this causal relationship. Right? All I can show you is this correlation. But I think based on what we see, there should be some calculation deficits in such patients if they have bilateral lesions. Yeah. So how about the coding of these neurons? Can we learn something else from this? What we did here is a cross-temporal decoding analysis with the classifier that I just told you, but now not simply in a large chunk of recordings, but the question here was if a classifier is trained and tested at the beginning of such a rule period, could it also predict, based on the firing of the population of neuron at the end of such a rule period, whether the patients would follow a calculation or a subtraction rule? In the hippocampus, this is what you see here. So this is the testing time and this is the training time. This is where about the rule cue has been shown. And these warm colors here in the square-like arrangement tells us that whenever classifier is trained at the beginning of a rule period, it can nicely predict also at the end of the rule period what calculation rule the patients are following. And this is giving rise to this square-like arrangement, indicating that we are dealing with sustained neurons that increase the firing rate and maintain an increased firing rate towards the end of the delay period. This looks very different in the parahippocampal cortex, where we only find this diagonal of high predictability, indicating that the neurons are only selectively tuned at a very specific moment in the delay period, but the tuning at this moment cannot be transferred or is not predictive towards the end of the period. So this is what I was just telling, sustained activity in the hippocampus. Neuron increases and is becoming selective at the beginning of the delay period and maintains the selectivity towards the end. And this is called a static coding in working memory, whereas neurons may turn out to be dynamic cells that are only very transiently encoding their preferred rule, Q, or their preferred rule, but don't do this towards the end. And this has been called a dynamic coding. And we think we find these two different working memory codes represented in these nearby areas in the medial temporal lobe. And then the question is, of course, what is their function? No one really knows. There are some speculations about the different functions. So one idea is that this static code is used for mental manipulation of attended items in working memory, which might be represented in the hippocampus, as opposed to the dynamic coding, which could be a reflection of a short-term memory code and not yet a working memory code. So in other words, these two areas could have different functions in maintaining arithmetic rules in working memory. And now yes?
I wanted to know the quantity of, like, the identity of the number, the identity of the quantity, is it expressed in, like, terms of the population? I mean, like the number of neurons activated, or?
So we have different neurons that are tuned to different numerosities or numbers, yes. I'm not entirely sure whether I understand your question.
No, I was trying to say, like, for example, the number 1 has like a super predefined, like, ensemble of neurons, for example.
You mean one dot and the numeral 1?
Yeah, like, for example, the neuron 2 and 3 respond to the quantity of 1. Are they selective only to 1, or they go for like 11, for example?
We didn't test here numerosities beyond 5. So this was only from 1 to 5.
Yeah,
I can't tell anything about higher numerosities. And we never went to 2-digit numbers because they are problematic, because you have 2 symbols for different numbers. Yep. So last animal species, our carrion crows. We have a little breeding colony in Tübingen. We wait until they're hatched. Then we take them into our human custody. We raise them. They become imprinted to us. They become very tame and interact with us. We train them many things, do them many things. We haven't succeeded yet to teach them reading. If you look closely, this paper is turned upside down, but they can do many other interesting things. And why are crows interesting for us? This is a very simplified tree of vertebrate life. And as I said before, birds and mammals belong to the amniotes, and the last common ancestor lived about 320 million years ago. And since then, the dinosaurs evolved, and birds are actually, zoologically speaking, the last descendants of the dinosaurs. So they are, in a very deep sense, reptiles. And you see the descendants here from the birds. And they are really this other type of vertebrate animal compared to primates and humans, but they are also extremely smart. And this is, from an evolutionary point of view, very interesting to us. This is also reflected in their brain, in their telencephalon, their end brain. It's very differently made up. And I very briefly show you some developmental neuroanatomy. So imagine we have an early embryo, a vertebrate embryo, and we make a cross-section through one telencephalic hemisphere. This would be a telencephalic ventricle, very early in embryology. And we would then look inside. We would find 2 different parts, the so-called pallium at the roof and the subpallium at the bottom. Subpallium is not interesting for me here, but what is interesting is the pallium, which is the mantle, basically. And you see, based on the 4 different colors, we have 4 different territories. And the interesting aspect is now what is happening with these 4 different territories. What we see in mammals and primates and us humans is that the dorsal pallium, the blue part, is becoming the major integration center in the telencephalon. The blue part is becoming the cerebral cortex, the area that makes us smart and conscious and what have you. Whereas in birds and reptiles, the ventral pallium, the green part here, is becoming the major integration center. It's a very different territory. So this is a classic case of convergent evolution, yet these birds are very smart, and I'll show you some examples later. In addition, the cerebral cortex is characterized by a nice 6-layered building plan, as you can see here, with some input and output systematics. This is very different in the bird brain, because in birds we do not find layered cerebral cortex. There is no cerebral cortex. Instead, birds have evolved these nuclear arrangements, and then every nucleus, that's one of the hypotheses, is functioning as one of these layers in the cerebral cortex, if you like. So a very different layout of the cerebral cortex. And of course, now we wonder, well, if everything is so different, do we at least find an area that is converging all the information in order to give rise to something that may function like the prefrontal cortex in the primate brain? So again, the prefrontal cortex receives multimodal information from all sensory modalities, and it is outputting information to the premotor and then to the motor cortex. It has high dopaminergic input from the midbrain, and so on and so forth. And we find, funny enough, an analog area in the bird brain that is called nidopallium caudolaterale, NCL here, this green part. This is based on a recent track tracing study where we followed the different connectivity patterns. This area in the bird brain, in the corvid brain, funny enough, sits at the back pole of the head, not at the front pole. But it does receive information from all secondary sensory areas, as you can see here. It has strong connections to the hippocampal formation. It has output to premotor structures. It has inputs to limbic systems, and it even has, and that's actually characterizing this area, strong dopaminergic input from the midbrain. So everything looks, functionally speaking, exactly like the prefrontal cortex, but the evolution is totally independent, and that's interesting for us. We train these crows on different numerosity tasks, similar to what I've shown you before in the monkeys, but now I'd like to switch to something more complex, which is a number production task. So here we were asking, can crows represent an instructed number by their own motor behaviors, and in this case, by pecking at the screen. So the task goes like this. The crow is facing a screen, then it is presented with an instruction stimulus, either a number of dots, or they have also learned previously to associate a number of dots, the value of the number of dots, with an Arabic numeral, the shape of an Arabic numeral 3. So here the crow should know that it has to peck 3 times. Then comes a motor planning period, a delay period, and then we present opportunities for the crows to make a single peck. So we instruct the crows very precisely when they are allowed to make a single peck in order to destroy timing information. Again, this is very important to us. We don't want the crows to do timing, we want them to really enumerate, to count if you like. And in this case, they would peck 3 times, and then also very important, at the end of the sequence, they have to tell us whether they are done with the counting sequence. So at the end of the sequence, they have to press what we call an enter key in order to tell us that now I'm done. Please evaluate and give me a reward when I was correct. And this is now done with numerosities and numbers from 1 to 5. Again, we are controlling, and I'm not going into the detail because of the advanced time, just to show you that we have 3 different temporal protocols that are only there to destroy temporal information, so that the crows can only rely on the number of pecks they are doing. And here is a video where you can see it. So you will see the instruction stimulus displayed here. Then there are these opportunities for the crows to peck a certain number, and then here at the bottom is the enter key that the animal has to press at the end of the counting sequence, and if it was a correct behavior, it will be rewarded from a feeder here down. So this is 3. 1, 2, 3, and then the enter key. So you see they're very enthusiastic and very focused. Now and again they do make errors, but I did not include the errors here in this movie. And while they're performing the task, very much like here, we can record from neurons in the nidopallium caudolaterale, this area that I just recorded from you in a tethered way, so the animals can still move around. They just cannot fly from the perches. And we find neurons that are nicely tuned to different impending numbers of movements. Here is a neuron that is tuned to 1 movement. Here is another one tuned to 3 impending movement. And here is a neuron tuned in the motor planning period to 5 impending movements. So before the animal is actually doing these counting pecks, the neurons indicate to us already how many they would do. This is at least what I'm claiming. We also tested this with a classifier approach, again feeding the entire population into our classifier, asking the classifier, can you predict based on these premotor responses how many pecks the bird would make? And this is the result here. This is the confusion matrix. And I hope you can see that most of the data, the accuracy values, lie on the diagonal. And this diagonal means that the predicted number was the same as the instructed number. Of course, this is not perfect, because now and again they make errors, and we don't only have a limited number of neurons. But I'm pretty sure if we have had all the neurons that the bird has available, all the data would be on the diagonal. And yes?
How many times does it take to train a crowd to do this?
How many?
Time does it take to train a crowd?
Many months, a year, something like that. Similar to the monkeys when you train them. So it takes a lot of patience, yes, for the students. But it's very rewarding if they can do it at the end. So this was indicative that the neurons are really meaningful, but as I told you, now and again they do make errors. They pack 1 fewer or 1 more. And if I'm claiming the neurons are meaningful, we should see these errors reflected in the neuronal responses. In other words, the neurons should respond differently whenever the bird is making an error. So we tested this with our classifier approach. First of all, the accuracy, so during the errors, is significantly reduced, is almost at baseline when we test for errors 1 smaller or 1 less compared to the correct. So these are the correct trials where the classifier is doing greatly. And then we also looked at the +1 or -1 error relative to a target numerosity. And the classifier can even predict, based on the neuronal responses, whether the bird is packing 1 too few or 1 too many relative to a target numerosity. And that was great for us to see, and that clearly indicates that what these neurons are doing is telling about the bird's behavior.
Sorry.
Yes?
Is there a nucleus that is like common between species?
Excuse me?
Is there a nucleus that responds like homogeneously in all of the species?
This is the first species that has been tested on such a task, as far as I know.
Okay.
We are now training monkeys. Actually, we have monkeys in the exact same task. The monkeys are not pecking, obviously, but they're making hand movements. And we also see beautiful data that we are currently analyzing. It looks very similar to the crows. Now, we had trained them now to do self-generated motor acts, but these birds are songbirds, as I told you. So we were wondering, could we train these birds to also utter a specific number of vocalizations? And that's the next task that I briefly show you. This is a bird now that is seeing a specific number, Arabic numeral here, and was trained to utter the same number of vocalizations, peck then somewhere here to tell us, I'm done now with counting, and give me a reward. So this is actually fantastic, for me at least. I think we would not be able to train a monkey to do this task. We have trained monkeys to vocalize on command, but this was only one vocalization. I guess this would simply be too much. So in terms of the behavioral data, and this is my last data slide, then I'm done. We were wondering, could we predict based on the acoustic features of the vocalizations? And by acoustic feature, I mean here the very first vocalization that the crow is doing in a sequence of vocalization, how many vocalizations the crow would do. For that, we resorted to these 23 predefined acoustic features, such as the temporal envelope, the spectral envelope, or the fundamental of the vocalization, and many more, 23. And we fed them into a clustering algorithm. In this case, we used these features for a classifier, to train a classifier, and then test the classifier with unseen data, with visual instruction stimuli, and you see based on a diagonal, the classifier has no problem, based on the acoustic feature of the first vocalization, to predict how many vocalizations the bird is going to make. And the bird was not only seeing visual instruction stimuli, but in other trials we also trained them to listen to arbitrary acoustic instruction trials, and also in an auditory feature, the classifier knows exactly how many vocalizations the bird is going to make. And if the classifier is trained on the auditory instruction stimulus, it can easily and significantly translate this information also to visual instruction stimuli, and vice versa. When the classifier is trained on visual instruction stimuli, 1 to 4 impending vocalizations, it can also transfer this information to auditory acoustic information. This is actually not under review, but has been published. Yes. And with that, I stop here. I hope I could show you that there are, in fact, numerosity-tuned neurons in the monkey brain, in the human brain, in the crow brain, that there are neurons that are very abstractly responding to calculation principles, such as greater than, less than, that monkeys can do. That in the human brain, we also found exactly the same code, tuned numerosity and number neurons represented in the medial temporal lobe, that these neurons are predictive and encoding calculation rules, such as addition and subtraction, that we find, however, 2 different working memory codes, a static code and a dynamic code represented in the parahippocampal and the hippocampus area. And finally, in the crows, where we also find exactly the same code, but here went a step further, and we're asking whether we can find signatures of impending self-generated counting movements, if you like, or counting vocalizations. And the answer is yes, that's something these birds can do. As I said, it's a lot of fun to work with crows because they are incredibly smart, extremely fast learners, and understand these general principles. In other words, have a high level of cognitive control, very similar to advanced non-human primates, and to some extent even to us. And with that, I stop. And thank you very much for your attention.
Hello, really, really nice talk. I was wondering what happens with the negative numbers. Is it the same to have 2 minus 1 or minus 1 plus 2?
Yes, the answer is no one knows. We have not tested negative numbers. We should do. I guess we will do in the next funding period. But so far, there are no data about negative numbers. It was crazy enough to investigate 0, particularly in animals, but now this was extremely rewarding. As I said, there's a lot of activity to the empty set, or in humans also to the number 0. And we need the 0 in order to transcend the countable natural numbers and to go to the negative numbers.
Thank you.
So this door is now opened.
So are these neurons that represent numerosity found in both hemispheres or just one
Yes,
hemisphere?
they are found in both hemispheres. In the human, in the animal studies and in the human studies, we do not find huge differences. We don't find any differences. And also based on the literature survey that I made a while ago, there's not really convincing evidence that there would be one hemisphere leading compared to the other. So my take is that both hemispheres are about equally involved.
Okay, thanks.
So I assume this is going to be a little bit of a speculative question because there might be no data for this. But if we imagine someone in finance, I don't know, imagine Elon Musk counting between 1.5 billion or 2.5. Do you think, I can imagine two options. One is that this billion is being transformed into just 1 or 2, or another option is that it's a completely different system for amounts, quantities like this. What would be your thoughts?
So again, here the short answer is I don't know the answer, but we are now talking about really something very different. I think a different representation of numerical quantities compared to what I showed you here. And I guess then it's very obvious that at that point we won't find this kind of tuned number neurons anymore. It's a different sort, a very symbolic representation of some sort. The human data, the functional data in humans indicate that also this kind of information will be represented in this parietal prefrontal number network. But how this looks at the level of a single neuron, I guess no one can tell, and certainly I cannot tell you. This is something that has to be explored, but it's a little nasty because you work then with these multi-digit numbers, and you have to disentangle the representation of every single value from the overall value of such a number. And that's something people shy away from because that's not trivial, particularly when we talk about billions of dollars or what have you.
Thank you.
Hi, I was wondering how are the crowds trained to produce these vocalizations? Because, I mean, I was curious about the process of the training.
Mm-hmm.
How do you make the crowd understand that they have to make a vocalization?
Yes.
And yeah, I was wondering.
Yeah, I'm sorry, this is top secret, I cannot tell you. No, I'm just kidding, I'm just kidding. As I told you, we hand raise them, so they make calls. They call a lot to us, initially begging calls. And we go from there. So when they get older and they call to us, we reward them. So they learn very quickly that vocalizing is good because they get a reward. This is the first step, and that's how we started. So vocalizing on command. We also could show that they can withhold a vocalization on command to a certain cue so that they're not vocalizing. So really, they have really excellent cognitive control over their vocalizations. And then you go from there. You just wait until they make 2 vocalizations, because maybe the first vocalization didn't get a reward, and then they try it again. And then you reward them for 2 vocalizations, and you keep going up to 4.
Okay.
They hate doing 4 vocalizations because it takes a long time, but fortunately they stayed with us for this publication.
Thank you, great talk.
But it takes a few months to get there, as you can imagine.
Oh, hello, great talk. I was wondering, was there a big difference in the accuracy for the crows when the instruction was circles than when there was an abstract number?
So this is something they first have to learn by heart. So we teach them to associate the shape of a numeral with a specific number of dots. And I'm not claiming that they are now symbolically counting. It's really just associating a visual shape with a numerical value. But after they have learned this, it doesn't matter to them, as you have seen. So they know exactly that the shape of a 4 indicates the value 4. And in fact, we have also published data from the Nidopallium caudolaterale, where we find many neurons that are tuned to specific numerical values, irrespective of whether it's a number of dots or these Arabic numerals that they've learned to associate.
Oh, thanks a lot.
Hello, I want to say thanks for your talk. First, I want to say perhaps there is no one that could answer this question. I only want to, I think it's interesting to debate on these ideas. And what I want to ask is, what do you think about how these individual differences in approximate number systems, proficiency correlate with symbolic maths, rapid lexical access, or general intelligence in humans.
Can you say the last part again? I didn't.
General intelligence in humans.
General intelligence. Oh yes, that's a good one. For all of these studies that I very briefly indicated, I did not show you the data, but for instance, the finding that the precision to discriminate set size is predictive of the children to do mathematics later in school, they have all been controlled for general intelligence. So it is not an issue of general intelligence that we are talking here. It's really a specific, if you like, a domain-specific mathematical capability that is investigated here, beyond general intelligence. Of course, it can be that general intelligence makes you do better mathematics, but this is then not the question. The question is there really something specific about mathematics? And in these studies, the answer is yes. There's a specific mathematical module. There's also some genetic indication that there's a genetic influence on mathematics capability, as there is for actually all behaviors. But it's not a fact of intelligence per se.
Thank you.
So, thank you very much, Andrea.
Thank you.
No se salieron.